Let's dive into a story that's like a high-stakes spy thriller, but it's all happening on a microscopic level. On one side, you have this pathogen that's so incredibly clever it only targets humans. And on the other, you have scientific ingenuity in a race against time to stop it. This is the story of the quest for a new typhoid vaccine.
So here's the central puzzle, right? I mean, imagine you're a scientist. You need to develop a weapon against an enemy, but you can't test it on any of the standard lab animals. Why? Because the enemy just completely ignores them. That is the exact problem researchers face with typhoidal salmonella.
So let's meet our antagonist here. This isn't just any old bacterium. It's a specialist. It's a highly evolved pathogen that has totally mastered the art of hiding in plain sight, inside the one animal it calls home, us.
Okay, so at its core, typhoidal salmonella is the bug that causes typhoid fever. And yeah, it's an ancient disease, spreading through the most basic things, food and water. But its method of attack is remarkably sophisticated.
And the scale of this problem? It is absolutely staggering. Globally, this bacterium affects nearly 22 million people every single year. This is not some rare disease. It's a persistent, worldwide health threat that really demands a better solution.
So how does this bacteria stay so hidden? Well, it's a master of espionage. First, it wraps itself in the special sugar coating, a via polysaccharide. Now that sounds complicated, but just think of polysaccharide as many sugars. It's a complex chain that acts like an invisibility cloak, helping it dodge our immune system's initial patrols. Then it gets even sneakier. It actively dampens the body's alarm bells, that inflammation that would normally signal an invasion. And this allows it to spread quietly and deeply throughout the body before our defenses even know there's a serious problem.
Now, given how stealthy this thing is, you'd think we'd have thrown everything we've got at it. But for decades, researchers have been stuck behind one major roadblock.
And this is it. This is the single biggest challenge. The bacteria is strictly host-restricted. It has tailored its entire existence to thrive inside the human body and absolutely nowhere else.
So what does host-restricted actually mean for the research? Well, it means you can't just infect a mouse or a rabbit and expect to see the same disease. The bacteria's unique set of tools, its keys to causing an infection, they only fit the locks on human cells. And this makes studying the disease and, you guessed it, testing new vaccines incredibly difficult.
So to get around this, scientists had to get really creative. They developed these workarounds, like using mice that are genetically modified to be more susceptible to human pathogens. And instead of the natural route of, say, swallowing the bacteria, they might inject it directly into the body. Now, these aren't perfect models, of course, but they allow researchers to mimic parts of the disease and test whether a potential vaccine can actually trigger a protective response.
And all of this has led to a really fascinating new idea for a vaccine. It's an approach that uses the ghost of the bacteria to teach our immune system how to fight the real thing.
And believe me, this innovation cannot come soon enough. The live vaccines we have now, while they work, they carry a small but very real risk of turning back into their dangerous, disease-causing form. Other types, they only offer temporary protection. And all the while, the bacteria itself is evolving. Drug-resistant strains are becoming more common. And different types, like Paratyphi A, are starting to cause a bigger share of the infections. And this brings us to this brand new concept, Outer Membrane Vesicles, or OMVs. Just think of them as these tiny, nano-sized bubbles that naturally pop off the bacteria's surface. These little bubbles are like harmless ghosts. They're not alive, they can't multiply, they can't make you sick, but, and this is crucial, they are covered in the exact same surface markers as the live bacteria. They basically present a perfect mugshot of the enemy to our immune cells, training them to recognize the real threat without any of the risk.
So the goal of this research was to use these OMVs to create what's called a bivalent vaccine. That's essentially a 2-for-1 shot. It contains the OMVs from both Salmonella typhi and that other increasingly common one, Salmonella Paratyphi A. This way, you're training the immune system to recognize and fight both of the major causes of enteric fever at the same time.
So the theory is brilliant, right? But does it actually work? Well, researchers set up an experiment in those special mice to find out.
Okay, so here was the game plan. The mice got a primary dose of this new OMV vaccine, and then they got 2 booster shots over the next 4 weeks. This schedule's designed to build up a really strong, layered immune memory. After the boosters, it was time for the final exam, challenging the mice with the live, dangerous bacteria.
And the results were just remarkable. This chart shows what happened when the bacteria were mixed with blood from both groups of mice. Let's focus on S. typhi first. See that really tall bar? In the blood from non-immunized mice, the bacteria population just exploded. But then, look at that short bar. In the blood from the vaccinated mice, the bacteria were stopped dead in their tracks. And the same exact pattern holds true for S. Paratyphi A. The growth was massively shut down for both.
The verdict was crystal clear. It worked. The antibodies and the immune cells that were generated by this OMV vaccine were incredibly effective at recognizing and neutralizing the real pathogens. The training paid off, big time.
This success really opens up a whole new chapter in the fight against typhoid, and you know what? Potentially against other stealthy pathogens as well. Just as the researchers themselves concluded, the changing face of this disease demands exactly this kind of approach. A bivalent vaccine isn't just a nice-to-have, it's becoming a necessity to provide broad protection against the different strains that are actually causing disease out there right now.
And this leaves us with a really powerful question for the future. This whole OMV platform, this idea of using the harmless ghosts of bacteria to train our bodies, it could be a total game changer. Could this be the key to finally developing vaccines against some of our most difficult and highly adapted pathogens? It's a really promising new frontier in our very, very old war against disease.